Types of ribosomes. Ribosomes are classified into two types based on their sedimentation coefficient, 70S and 80S. S stands for Svedberg unit and related to sedimentation rate. Sedimentation depends on mass and size. Thus, the value before S indicates size of ribosome. 70S ribosomes. Prokaryotes have 70S ribosomes. The 70S ribosomes are comparatively smaller in size and have sedimentation coefficient 70S with molecular weight 2.7 times 106 Daltons. Electron microscopy measures the dimension of the 70S ribosomes as 170 times 170 times 200 Al. They occur in the prokaryotic cells of the blue-green algae and bacteria and also in mitochondria and chloroplasts of eukaryotic cells. 80S ribosomes. Eukaryotes have 80S ribosomes. The 80S ribosomes have sedimentation coefficient of 80S and molecular weight 40 times 106 Daltons. The 80S ribosomes occur in eukaryotic cells of the plants and animals. The ribosomes of mitochondria and chloroplasts are always smaller than 80S cytoplasmic ribosomes and are comparable to prokaryotic ribosomes in both size and sensitivity to antibiotics. However, their sedimentation values vary in different phyla. 77S in mitochondria of fungi, 60S in mitochondria of mammals and 60S in mitochondria of animals. The ribosomes are chemically composed of RNA and proteins as their major constituents, both occurring approximately in equal proportions in smaller as well as larger subunit. The 70S ribosomes contain more RNA, 60 to 40%, than the proteins, 36 to 37%. While the 80S ribosomes contain less RNA, 40 to 44%, than the proteins, 60 to 56%. There is no lipid content in ribosomes. Ribosomal RNAs. RNA constitutes about 60% of the mass of a ribosome. The 70S ribosomes contain three types of rRNA, viz. 23 srRNA, 16 srRNA, 5 srRNA. The 23S and 5 srRNA occur in the larger 50S ribosomal subunit, while the 16 srRNA occurs in the smaller 30S ribosomal subunit. The 80S ribosomes contain four types of rRNA, 28 srRNA, or 25 to 26 rRNA in plants, fungi and protozoa, 18 srRNA, 5 srRNA and 5.8 srRNA. The 28S, 5S and 5.8 srRNAs occur in the larger 60S ribosomal subunit, while the 18 srRNA occurs in the smaller 40S ribosomal subunit. About 60% of the rRNA is helical, i.e., double-stranded, and contains paired bases. These double-stranded regions are due to hairpin loops between complementary regions of the linear molecule. Ribosomal proteins. A ribosome is composed of 3, in bacteria, or 4, in eukaryotes, different rRNA molecules and as many as 83 proteins, organized into a large subunit and a small subunit. The primary structure of several of these proteins has been elucidated. Most of the recent knowledge about the structure of ribosomal proteins has been achieved by dissociation of ribosomal subunits into their component rRNA and protein molecules. When both 50S and 30S ribosomal subunits are dissociated by centrifuging both of them in a gradient of 5m cesium chloride, then there are two inactive core particles, 40S and 23S, respectively, which contain the RNA and some proteins called core proteins, CP. At the same time, several other proteins, the so-called split proteins, SP, are released from each particle. There are SP50 and SP30 proteins which may reconstitute the functional ribosomal subunit when added to their corresponding core. Some of the split proteins are apparently specific for each ribosomal subunit. The split proteins have been further fractionated and divided into acidic, A, and basic, B, proteins. Metallic ions. The most important low molecular weight components of ribosomes are the divalent metallic ions such as Mg++, Ca++, and Mn++.